Hello everyone, this is Dr. Ali Mohammad Maulali from uh, Thailand. I am a faculty at School of Food Industry, King Mongot Institute of Technology, Lakraba. And today's presentation, my topic is on sustainable valorization of seafood and poultry processing byproducts. And mainly, I will be speaking about two byproducts, fish liver from fish processing industry waste, and poultry from poultry. I'll be considering chicken gizzard. So chicken gizzard, mainly we have fermented the chicken gizzard using Pediococcus acidilactaceae. And why we have selected this is mainly because of two reasons. One is the higher protein content, and second is mainly the iron, significant content of iron. And as we can see, chicken gizzard is mainly rejected by many people because of its higher microbial load and because of hard texture, so it is difficult to consume by most of the people, and it has been rejected by many people to be consumed or to be used in a regular diet. So here we have tried to use Pediococcus acidilactaceae to ferment and extract the protein hydrolysate, and we have also determined the effect of fermentation on the other significant properties, a change in the properties such as retention of free amino acids, uh, fatty acids, as well as iron content. So here I will mainly discuss on the brief on the fermentation aspects, and I will not go deep in the topic, so there is a limited time frame. And as we see, the processing waste chicken gizzard, it contains most of the fatty tissue and other debris and undigested matter of food matter which is left over in the gizzard. And we remove all these residues, and we mainly retain the intact the tissue, the gizzard, as we can see here. This is then minced to make paste, and later on we steam cook at 80 degrees to remove the and deactivate enzymes, or to loosen the tissue, etc., for before fermentation. And during fermentation, we can see we have separated the hydrolysate, and we have collected the hydrolysate to determine various properties. And residue, we have also collected the residue to check various properties with similar to that of hydrolysate. And here we found the suitability of Pediococcus acidilactaceae, and we found this ATCC 8042, a potential culture that has higher amount of proteolytic activity, and we found a significantly lower lipolytic activity. And this was very good in our observation, as we can retain many fatty acids during fermentation. As we can see, the trend in change in pH and the change in the trend in total titratable acidity indicated that the fermentation took place in a significant amount, and this was confirmed by two other factors: degree of hydrolysis and extractability. As we could see, we have fermented for 24 hours, and we could see that the degree of hydrolysis and extractability of protein hydrolysate was significantly higher at the 24 hours. So it was almost to the 80%, of the extractability was up to 80%. This was done based on the protein content and through microgeldal method. So next, as we know, this gizzard contained higher amount of microbial load, and we found the total viable count was significantly higher, as you can see on the left hand side, the log units, and the total viable count was significantly higher at the initial, and uh, uh, you can see the mostly uh, microbes that present in the gizzard was lactic acid bacteria, E. coli, staph, and clostridium. So these, during fermentation, as we can see, we have added some lactic acid bacteria, Pediococcus acidilactici, which during fermentation increased the number and log CFU. And you can see relatively there was a significant reduction in the pathogenic strains, and we could see first elimination was from E. coli, later on Clostridium, as well as staph. So this during 24 hours we could not detect any pathogenic strain. This indicated that the Pediococcus acidilactici also had a significant antimicrobial property, which deactivated or these microorganisms. And we have separated the um, residue as well as the protein hydrolysate, and we have studied various extract, and mainly the antioxidant activity was determined in the hydrolysate fraction, and we could found that the increase in the amount of antioxidant activity was relatively higher with respect to the fermentation time. And we also found the activity, antioxidant activity, such as DPPH, ABTS, and FRAP were relatively dependent on the type of solvent we have extracted the peptides. So a few peptides such as water extracted in water were demonstrated lower activity for DPPH, while they demonstrated higher activity in ABTS, and you can see there is lower activity in FRAP. And you can see the activity of FRAP was relatively almost similar to both of the extracts, while we can see these the activity of water and ethanol was significantly different in the ABTS and DPPH, and the activity we found the region where the plateau was reached at 18 hours in most of the antioxidant activity, and later on the stability or decrease in the activity was noticed after during 24 hours. So next, what we have determined was the total iron. As we know, gizzard is one of the richest source of iron content, and we have determined based on the dry weight, and we could see the iron content based on milligrams per kg of sample. And in the raw minced gizzard, you can see the total iron content was very high, and relatively the free iron content was soluble iron content was lower. So this indicated that the soluble iron content could increase during fermentation, as we can see in raw minced and minced cooked. You can see during cooking process there was very lower soluble iron content, and during fermentation the soluble iron content increased significantly, and in the residue we can see the lower iron content in the residue. This indicated that the protein hydrolysate was the rich uh, with peptides as well as with the soluble iron content. So this indicated that the fermentation did not affect the iron content, though we could see the a little bit change in total iron. So this was distributed among the fermented hydrolysate as well as the residue, but the overall total iron content was almost to the maximum level. Okay. Next, we found the essential amino acids as well, which were not detected in the free form. So mainly this is the free amino acid composition, and we have found that this there are several amino acids which were in the free form released in the protein hydrolysate, and we found these several were classified as essential and non-essential, and several amino acids which were responsible for antioxidant activity were also significantly released at the higher amount, higher content. Okay, so you can see the total essential amino acids, we could see this much higher increase as with compared to the raw sample. This indicated that the higher, very high amount of hydrolysis mechanism of the pediococcus acidilacticide. So this culture was highly suitable for fermentation of chicken gizzard. And next we found the fatty acid profile, which was uh, uh, which indicated that the uh, unsaturated fatty acids, including monounsaturated fatty acid, polyunsaturated fatty acids, were retained to maximum extent, and indicating that the culture had lower lipolytic activity. So this was highly suitable for fermentation and for retaining the property. And in addition to this, the retention of the unsaturated fatty acid indicated that the peptides as well as amino acids have helped in retention of these amino acids from oxidation. Okay, so overall conclusion, this study highlighted the potential importance of protein hydrolysate that could be produced from chicken gizzard as an excellent source of bioactive peptides and amino acids, as well as the significant amount of iron, soluble iron content. And the fermented chicken gizzard protein hydrolysate showed potential antagonistic property against gram positive as well as gram negative, as we saw in the graphs. And the hydrolysate exhibited significantly higher amount of antioxidant activity. This can also be related to the free amino acid as well as peptides rich in hydrophobic amino acids. Next, the fermentation using pediococcus acidilacticase, especially ATCC 8042, facilitated the conversion of soluble and heme iron, so which is very important for the as a bio available iron, which can be used as a supplement for humans, which humans for improving their health or improving the iron content in their body. And these observations indicated that this fermentation of pediococcus acidilactase could be important for retention of antioxidant activity as well as retention of iron content, and as a potential which this could be a potential additive for development of any functional food to reduce oxidative stress and to treat iron deficiency. So this was a very successful study, and we have published this literature already. You can see the title.
Okay, we will hear our next speaker for this session. We will have our presenter, Dr. Dede Heri Yulianto from Research Center for Applied Microbiology, National Research and Innovation Agency, BRIN, Indonesia. The title will be Microbial Technology for Green and Sustainable Environment in the Case of Textile Dyes Wastewater Treatment. Please, we can hear the presentation from Dr. Dede.
Thank you very much. I would like to introduce myself. My name is Dede Heri Yulianto. I'm from Research Center for Applied Microbiology, National Research and Innovation Agency. In this occasion, I would like to present our research with the title Microbial Technology for Green and Sustainable Environment in the case of textile dye wastewater treatment. In Indonesia, around 85% of the river has been polluted and mostly by the textile dye wastewater. It is estimated around 200,000 tons of dye starch is expelled into the global environment every year. The textile dye are originally designed to be recalcitrant for long-term use. It is usually contain anthraquinone and azo dyes bond, and it is resistant to aerobic wastewater treatment and therefore making an environmental problems. The textile dye wastewater is toxic, can be irritated to the skin and eye, and mutagenic and also carcinogenic. There are several treatments for textile dye wastewater, such as physical methods using absorption, membrane filtration, and also coagulation, and also using chemical methods like advanced oxidation using zero valent aluminum, and also penton reaction or ozonization. By using biological method, sometimes bioremediation of textile dye can be used by using fungi or bacteria or, and also their enzyme to degrade some textile dyes into non-toxic compound. There are limitations of different techniques. Chemical methods are simple, rapid, and efficient process, but it is economically not feasible for small and medium enterprise like batik community in Indonesia. And also membrane filtration is rapid, but it high cost because it need the operational cost. And also for coagulation and flocculation is a simple physicochemical process, but will generate sludge and increase process cost. This is some of the uh, process. I describe how the process of the coagulation and flocculation is a simple method, but that will generate sludge in the end of the process and need to be processed and increase the operational cost like this. So, according to our study, we found that efficient treatment can be used using the absorption method because it is easy, simple, flexible, and operative in the wastewater treatment plant, like in the small and medium enterprise in Indonesia. However, absorption method is only move the toxic compound into the material, but cannot degrade the chemical into non-toxic compound. In other hand, microbial degradation using the fungi is a simple and economically attractive technique. It also can degrade toxic compound into non-toxic compound, but the process is slow, needs 6 until 30 days. So to solve this limitation, we have to find supermicrobe to degrade fastly the toxic textile dyes, or we use immobilization or combination with the absorbent. In this process, the absorbent will absorb the dyes and the fungal will degrade the dyes into the non-toxic compound. Enzymatic degradation also over the past process in hours, but it is challenging how to produce low-cost enzyme. We can solve this problem by finding novel enzyme or by using the recombinant, or we use immobilization or combination with the absorbent. In this study, I will present my research in how use this fungal microbial to immobilization with the absorbent to enhance the degradation and also to degrade the toxic compound into non-toxic compound. So during 2014 until now, I have we have screened, isolated, and screened the potential fungi for degradation of dyes. We conducted the degradation in liquid medium by fungi and enzymatic degradation, and we developed several immobilization of mycelium and enzyme on the biomaterial to improve the stability, stability of functional stability, and also storage stability. This is the key technology for the green and sustainable wastewater treatment. So how can water fungi degrade dyes? Water fungi can degrade the lignin component in wood by producing some ligninolytic enzymes such as manganese peroxidase, lignin peroxidase, and also lacase. The lignin compounds are similar with several dyes compound. Therefore, some ligninolytic enzyme like lacase can also degrade the dyes in this process. So we isolate and screen several places in Indonesia during 2015, like in the National Park of Laiwangi and Wangameti, and also in Gandang Dewata, in Taman Eden 100 in North Sumatra, and also in Bubak Sembilang National Park by using the MRB UNESCO program in 2019 and other places. So this is the so we isolate and screen the potential fungi in the lake by using the collective fungi in the by using the growing the fungi in selective agar medium containing Zapekdok and lignin agar medium. The fungi will the fungi can degrade lignin will demonstrate it red brownish color here, and then we purify on malestaf agar medium, and then we screen on the lignin and FBBR agar medium. The decolorization ability of the fungi can show when they decolorize the color into the colorless compound. So we calculated the ghost diameter and also the decolorization diameter of the fungi, and we found that Tametacisuta D7 have the high ability to ghost and decolorize by using this textile dye as the carbon source of the ghost. And we identify the fungi using the DNA sequence analysis and comparing by the mushroom DNA extraction and also the mycelial DNA extraction, and we identify the fungi using this method. And we found that the fungi can decolorize some LBBR dyes, this is anthraquinone dyes, and also AB129, but this is different in the chains of the backbone of the dyes, and also this is the AB5 containing 2 azobone. And some dyes can decolorize rapidly, this all the dyes, like Thymetes yesuta and also Thymetes ADN 82. So we biodegrade and analysis of the metabolic pathway during the anthraquinone degradation by Thymetes yesuta D7 immobilized in like expanded aggregate and toxicity assessment. So the first, we immobilize the fungi in the leca. So leca is a stone ball usually used in the plantation. So we steam activated of this leca and washing to remove several impurities in the leca, and we immobilize fungi in leca, and we obtain this mycoleca. We call this is mycoleca because the leca containing the myco. And according to same analysis, the mycelia fungus can penetrate it into the whole of the leca. As we can see in A figure is the only leca, and the B figure is leca containing the mycelia from the Thymetes yesuta D7. And we use this LECA to decolorize BBR, B4, and also AB129. These all dyes are anthraquinone dyes. And we found that the microLECA can decolorize all the dyes attractively during 12 hours or 24 hours. And also according to the uh, kinetic reaction, LBBR is mostly easily degraded compared to other dyes like AB4 and AB129. And uh, we also measure the detoxification after degradation using the human dermal fibroblast cells because this cell line has been frequently used to assess cytotoxicity, skin cancer, skin irritation, and also mutagenic caused by the environmental pollutant. From this result, we found that LBBR reduced the cell permeability around 0.6%, and also AB129 and also AB4. But after uh, after degradation by the microLECA, the cell permeability increased and also almost non-significantly different with the control non-treatment. So it saw detoxification by the microLECA. And this is we analyze the metabolic pathway of anthraquinone degradation by the microLECA. This is LB4, LBBR, and also this is the AB129. We found that the LECA, the lacase enzyme produced by the fungi in the microLECA, can degrade the no, it can degrade here the R1 and also NH bond and produce small component. And this small component is non-toxic compound compared than the parent dyes. The other, we also investigating the biodegradation of azo dyes by the Tametensis-Uta-D7, and we analyze using UPLC FTICR-MS supported by in silico simulation and toxicity assay. So this is the water fungi can produce extracellular enzyme and degrade some azo dyes, and the degradation we analyze using the UPLC ultra high performance uh, mass spectroscopy, and also we analyze the meta degradation pathway and cytotoxicity and also genotoxicity. We found that microLECA also can degrade all the azo dyes test in study like ABBR, AB113, and also AL7 with the different degradation ability. And we also identify the detoxification of this Tametes Hinsuta treatment to the dyes, and we found that LB5 reduced the 42% of the cell permeability, but after treated by the microLECA, the cell permeability increased. Also, by using in human dermal fibroblasts, we can see that after treatment by the microLECA, the almost 100% of the cell permeability has been observed in the reaction. And this is the metabolic pathway of the AB5 degradation. We can see here the parent component of AB5 is decreasing during the degradation time, and we found so many metabolic degradation here, and we analyzed the root of the megadegradation pathway, and we found that this fungi can effectively degrade and detoxify the parent component of the AB5 into small component. And also this is for AB113 degradation, and this is acid orange 7 degradation by my colleague here. And the fungal immobilization on LECA also have the excellent reusability because after we use for decolonization in Purwokerto it is still can be used until several times, until more than 25 times, and the lacase activity in the uh, culture also still detected. Also, this is in the Pakalongan printing batik, and we can see that the lacase activity still occur during the degradation, and the microlaca can be used more than 25 times. And this is the microlaca treatment in the printing batik. We can see that the degradation after 42 hours, the light is higher compared than before. And this is the degradation after 24 hours. And we use the continuous system until 3 weeks. And the plant is now better if we use this mycoleca. And there are natural pH and also decreasing the COD level and also decolorization. And we analyze this in the wastewater whether the enzyme activity still produced or not during the degradation. And we found that after addition of ABTS, the color changed into the green, and it is a positive containing the enzymatic activity. And this is also microleca in textile dyes wastewater. In Jakarta, it can decolorize 75% and reduce COD, phosphate, and also ammoniac, and also the total chromium, and reduce the toxicity until 83%. So we conclude that this study clearly showed that the microbial immobilization in LECA has the potential for green and sustainable environment, particularly in textile dye wastewater treatment. We acknowledge our research group, Composite Engineering Research Group, and also the National Research and Innovation Agency, BRIN, Kyoto University, Japan, Kyungpook National University, Republic of Korea, Chulalongkorn University, Thailand, and also University of Science, Malaysia. Thank you very much for your kind attention.
Okay, thank you very much. We will continue for the next presenter. For the next presenter, we will have our Associate Professor, Dr. Asusuke Yoshida from Nara Institute of Science and Technology, NAIS, Institute for Research Initiative Division for Research Strategy, Division of Biology Science, Nara, Japan. And the title will be Understanding a Microbial Polyethylene Terephthalate, a PET metabolism and its application for biodegradable plastic production. So we can hear now the presentation, please.
I thank you all the organizing committee members for giving me such a great opportunity to present our work. I am Shosuke Yoshida from Nara Institute of Science and Technology. And today's talk title is Understanding a Microbial Polyethylene Terephthalate PET Metabolism and its Application for the Biodegradable Plastic Production. We produce plastics from petrol. They are partially recycled, but most of them are incinerated, landfilled, leaked into the environment after use. So the problem is that the non-biodegradable property of plastics burdens the environment. So far, few plastic degraders have been isolated, but once identified, understanding their mechanism could also be applied to create a sustainable economy and environment. Let me introduce one of the non-biodegradable plastics, polyethylene terephthalate, PET. PET is used as packaging materials and fibers, and PET can be synthesized from ethylene glycol, EG, and terephthalic acid, TPA. Therefore, PET has ester linkages between the monomers, and PET is non-biodegradable. Is known to be non-biodegradable. but we know that a lot of and a variety of esterases in nature, so we expected that very few, very unique esterases could hydrolyze PET. So we searched for the microbes that degrade PET, and recently we have successfully isolated the PET degrader and give us new species name with Ideonella saccharensis 201F6, and this bacterium not only degrade PET but also assimilates PET. This is a scanning electron microscopic image of this microbe. This is a single cell and the cells on PET. By removing the cells from the PET film surface, you can observe the degraded traces of PET film surface. You can compare with the control. PET hydrolytic enzymes, we say PHEs, have been reported since 2005, and most of the enzymes are cucinase homolog. And what is cucinase? The cucinase is an enzyme that hydrolyzes cucin, found in such as the plant leaf surface. And the structure of cutin is aliphatic polyester with branches. On the other hand, PET is aliphatic and aromatic polyester. We have sequenced the genome of isaac and found only one open reading frame coding for the cucinase homolog here. So we have produced the recombinant protein of this gene and incubated the proteins, the purified proteins, with an amorphous PET at very mild condition, actually 30 degrees Celsius and pH 7.0 for 18 hours. As a result, this protein produced many cavities at the PET film surface like this, and water-soluble compounds such as TPA and MHET, which is monohydroxyethyl terephthalic acid, composed of one ethylene glycol and one terephthalic acid. This is actually, we can say that the minimum unit of PET polymer, because MHET repeats produce the PET polymer. So anyway, so these TPA, MHET, water-soluble compounds has been released into the reaction buffer. So we can say that this enzyme from iodine is PET hydrolytic enzyme. Okay. So this is a phylogenetic tree of non-PET hydrolytic enzymes. To identify the difference between the iodine enzyme and other PHEs, we chose 4 different types of enzymes. One from iodine one from Fusarium solani, which is fungus, one from Samobifida fusca, which is a thermophilic actinomycetes, yep, and one from uncultured bacterium. Then we compared the hydrolytic activities of these 4 enzymes towards aliphatic esters, actually synthetic aliphatic esters, and their uh, substrates used for the typical substrates used for characterization of cutinase, and also uh, we measured the activity towards PET film, which is aromatic aliphatic polyester. Surprisingly, I-saccharinaceous enzyme has exhibited much lower activities against aliphatic esters compared to these three other pHs. But in contrast, iduronsaccharinases enzyme exhibited much higher activities against PET film, compared to these three pHs. From this extremely high PT hydrolysis activity and PT specificity, we designated this enzyme as PT hydrolase or PTS. This is superimposition around catalytic site of PTS structure with another type of PT hydrolytic enzyme, LCC, this white uh, ribbon representation. Uh, the most of the residues around the active site are identical, but histidine in LCC is replaced by tryptophan in PTS, and phenylalanine in LCC is replaced by serine in PTS. So we have replaced these two residues between PTS and LCC and measure the PT specificity of these mutant proteins. A mutant protein, PTS mutant, actually double mutant decreased PT specificity to 23% compared to the wild type of PTS. In contrast, LCC double mutant dramatically increased the PT specificity by 11-fold. From these results, tryptophan 159 and then serine 238 residues in PTS are involved in the very high PT specificity of PTS. As I showed you in the previous slide, PTS hydrolyzes PT and release MHT as a main product. However, PTS did not show MHT hydrolytic activity. And although MHT is a esteric compound, and also we know that isokinesis has both TPA and EG metabolism. These facts indicated that isokinesis has MHT hydrolase for the complete PT metabolism. So we searched for the gene coding MHT hydrolytic enzymes. We have cultured the cells on maltose as a negative control, and TPA and BHT, which is PT oligomer, and carried out transcriptome analysis. These magenta bars indicate the expression level of PTS gene. We found that one open reading frame shows a very similar expression pattern with PTS, so the blue ones. Also, this gene product is a putative tannase family protein known to hydrolyze the ester linkage of aromatic compound. Yes, as you noticed, MHT is also the aromatic compound having ester linkage. So we created the recombinant proteins of this gene and then measured the catalytic activity for various potential substrates. So this tannase family protein showed a significant MHT hydrolytic activity. The turnover rate to MHT was 31 per second, and the Km value was 7 micromolar, which is very high affinity between MHT and then the enzyme. So this enzyme was not active on PET, not very active on BAGT, and not active for several aliphatic esters and then aromatic esters. So we can say that this enzyme is very active for MAGT hydrolysis and distinguishes MAGT at high level, so that we designated this enzyme as the MAGTase. So next, we developed a gene disruption system of isokinesis and successfully knocked out PTS gene and MAGTS gene. The host strain for the gene knockout showed significant growth on PET. However, delta PTS and then delta MAGTS strains abolished the growth capacity on PET. So this indicated that PTS and MAGTS both are critical enzymes to utilize PET for the growth of isokinesis. And we constructed the metabolic model, actually PET metabolic model. IS cells, iduronate cells, secrete the PTS proteins to hydrolyze PT into MAGT molecules, then transport it into the periplasmic space. The second enzyme, MAGT, hydrolyzes MAGT into ethylene glycol and then TPA. These PET monomers are further metabolized by iduronate. This is a PET metabolic pathway of isokinesis. Putative enzymes necessary for ethylene glycol and then terephthalic acid TPA metabolism are coded on the isokinesis genome. Further, as an application of PET metabolism of isokinesis, we were interested in the polyhydroxyalkanoate or PHA production pathway from acetyl-CoA to PHA, because PHA can be utilized as biodegradable plastic. This is a PHA synthetic gene cluster in isokinesis formed with Far-C, Far-A, and Far-B, Far-C, Far-A, Far-B, and coding for PHA synthase, beta-ketosiorase, and acetoacetyl-CoA reductase, respectively. So they are highly homologous to the gene cluster in cupriabdes negator, which is a representative PHA producer. Therefore, we grew the isokine cells on PT and found that this bacterium also accumulates a large amount of PHA in the cell. That is, isokinesis can convert PET, which is non-biodegradable plastic, into biodegradable plastic PHA. And so far, 2 reported PHA production systems using PET as a fermentation feedstock have been reported, suggested 2-step process. The first step is pyrolysis or enzymatic PET hydrolysis, and the second step is the conversion of the produced monomers into PHA by microbial fermentation. They use the Pseudomonas. In contrast, our technology is able to do both hydrolyze PET and metabolize the monomers. This is a summary of today's talk. Thank you very much.
Okay, thank you very much, Dr. Yoshida. So we will come to our question and answer or discussion for this session. So we have our speaker here, Dr. Dede, Dr. Susiko Yoshida with us, but unfortunately we don't have Dr. Ali with us now, but you can deliver your question to Dr. Dede and Dr. Sosuke Yoshida in the chat box, so I can read the question and deliver to our speaker. So for I have first a question from Chandra Setiadi. I think this is from Indonesia. So for Dr. Yoshida, what parameters are possible to hinder the production of PETase and MHETase, or what parameter to enhance the production of those enzymes? Please, Dr. Yoshida, you can answer the question. Maybe you can unmute your mic. Oh, yeah.
Yeah, thank you very much for the question. So the question is, what is it possible? So what do you mean? I haven't get the point of the questions. So what parameters are possible to hinder the production of PTS and MHTS?
So because the PTS is useful to degrade the PT, yeah.
Mm-hmm.
And how we can enhance the production of those enzymes.
Enhance.
Yeah, because you make it recombinantly, yeah? Or it's
Oh no. So okay. So it's about the in the recombinant or the native. So actually if we use the host strain, I mean the ideal sequences. So actually to increase the expression of PTS and MHTS, we usually culture the cell with PET, because in the how to say,
so it triggered or induced, yeah.
Yeah, induced, and then
Yeah.
actually the expression is extremely high, and actually maybe those expression level of PTS and MHT is maybe within the top 10 expression level, sometimes top 1, top 2, something like that. And yeah, is it so? Yes, so induced, yeah, true.
Yeah.
So
just induce the culture with the PT in the culture, yeah.
Yeah, especially for MHTS, but PTS is how to say, it's so PTS is always expressing very high level, but yeah, for sure, the PT will also enhance the expression level.
And for the application in the field, do you think that we can directly use the microbe, the Adenolella sacchensis, directly to the waste, or we should isolate the enzyme?
Yeah, I'm thinking both. So utilize the recombinant enzymes. Yeah, if we need the proteins, just proteins. I mean, if you have a PTS and also the MHTS, you can hydrolyze the PET into the monomers. So I think in this case, maybe we should use the just proteins. But as I presented at the end of my presentation, we want to convert the PET into the variable compounds. So in this case, we have to use the ideal sacchensis, because the ideal sacchensis is the only microorganisms that has a complete PET metabolism. So the two options in my mind.
Okay, thank you. Maybe for the next question, I will deliver the question to Dr. Dede. Hello, Dr. Dede. I would like to ask you a question, maybe regarding for your presentation. It is very, very interesting that you can find the case, yeah, for degraded the waste. Maybe my question also a bit similar to Dr. Sasuke Yoshida. If we want to apply this technology or the finding that you have in your research to how to say, to solve the problem for the waste in a field, yeah, how can we apply it? Because maybe it will be different. If it in industry, we can use like a big container, yeah. But if it the waste is already in the environment, for example in the lake, yeah, or in the river, how can we apply this technology or the enzyme or the microbe that you already found to solve the problem?
Okay, thank you very much, Bu Chatur, for the question. Yeah, actually, we can improve the stability and also, yeah, stability of the microbe or enzyme by using the immobilization in the a very good material. And we found that LICA, like expanded clay aggregate, can be used as a supporting material of the immobilization of this microbe. And we also already test about the viability this leca, this fungi in leca, and also we have directly test in the wastewater treatment, particularly in textile or batik dyes wastewater in Pekalongan. And also, yes, it is true that the composition of the wastewater actually is not uh, as easy as like we use pure chemicals dyes. So we found that some, for example, batik wastewater is usually they contain like a wax or other compound. It is very inhibit the ability or capability of the fungi itself. So in my proposal, we try this mycolica in some part of the step in the wastewater after uh, like a coagulation or yeah, precipitation, some solids material, then only the color stay in the wastewater, then we can apply. So in application, we can use like a bioreactor system by using the, for example, we use the uh, rotating biological contactor by putting the mycoleca in the drum, and then we rotated this LB rotating drum, rotating biological LB rotating drum biological contactor, LDBC. This LDBC we use, and we observe above the quality of the wastewater. Yeah, by using this immobilization, I think we can. But actually, we still trying in some part in Pekalongan, yeah, by using this technology. I think that's all for me. Thank you
Okay,
very much.
that's very interesting. Yeah. So so we can use or have a benefit from this microbe for solve the problem with the waste, especially with the dye. And for Dr. Susuka, we can deal with the plastic waste. Yeah. So I think for our session, we will have if there is more question maybe from the audience. If there is no more question, I think we will have a small break. We will also to have the award for our presenter. From the committee, there is a certificate will be displayed in our Zoom meeting. Maybe from the committee, you can display the A certificate for our presenter. So the first one for Dr. Dede, maybe. There is like time, sorry. Please wait for from the committee. Can you display the A certificate? Okay, if there is some technical problem, I think we can send our certificate to our presenter by email. So once again, I will say thank you for our speaker, our presenter, Dr. Dede, and also Professor Sosuke Yoshida for your nice presentation. And for all the audience, we will have a small break. We will join again this session in the next, I think in Indonesia time is at 2:30. Here. So we have the certificate. So this is for Dr. Ali Muhammad Maula Ali. The certificate from the committee. We say thank you for the your opportunity to present us or sharing us with your research. And the next one maybe for Dr. Dede Heri Yulianto. Thank you.
Thank you very much.
Yeah. Thank you once again. And then the next one for Dr. Sasuke Yoshida. Thank you very much for your sharing session, that very insightful. So for all participants, we will have small break. We will continue again in Japan time in 4:30. Okay, everybody. Okay, welcome back to our second session. We are now in joint SBT meeting with Indonesia, Philippines, and Thailand. In this session, environmental biotechnology, for the second session, we will have 3 speakers from Philippines, from Thailand, and also, the last one also from Philippines. So today, without any further ado, we will present our first speaker. I think Dr. Jessica Simbahan already with us. So we will hear her presentation. The presentation title is Bioconcrete, the Future and the Construction Industry. Dr. Jessica Simbahan from University of Philippines, Diliman. And for no further ado, we will hear the presentation from Dr. Jessica Simbahan. Please.
Good afternoon, everyone. Today I will be presenting my talk on bioconcrete, the future of the construction industry. Concrete is the single most widely used material in the world. It is durable, cost-efficient, and its materials are locally available. It is simple with only 3 ingredients: aggregates, water, and cement. However, its key component, cement, contributes to 80% of total greenhouse gas emissions of concrete. Cement releases about 2 billion tons of carbon dioxide into the atmosphere per year due to its production, which uses high amounts of energy to obtain calcium from limestone. Its production accounts for 8% of yearly global carbon emissions. This makes cement production highly unsustainable due to its to that. The geological formations in caves are produced abiotically when rainwater passes through the soil and absorbs carbon dioxide, forming carbonic acid, which helps to dissolve underlying rock. When this water seeps into the air-filled cave, it loses carbon dioxide to the cave atmosphere, causing the water to precipitate calcite deposits in different forms. Aside from this process, microorganisms have also been speculated to contribute to these geological formations. Their metabolic activity influences the microenvironment, favoring precipitation of calcium carbonate, with the cells acting as nucleation sites for crystal formation. This process can be harnessed through a technology known as microbially induced calcite precipitation, or MICP. Biocement produced by MICP is a new and emerging technology in the construction industry. This technology can be used to produce bacterial bricks. Unlike cement, MICP technology is a natural biological process which can be accelerated in the laboratory. It has low energy input since it can be produced at room temperature, and it has a low carbon footprint. Certain bacteria have the capacity for MICP. The negatively charged cell walls and exopolysaccharide of the cells attract calcium ions from the surroundings. The bacteria then obtain their energy by secreting urease, which converts urea in the environment to ammonia and carbonates. The ammonia increases the pH of the surroundings, and the carbonates bind with the calcium ions, forming calcium carbonate crystals, which bind the sand particles together. In order to isolate bacteria capable of MICP, we conducted our sampling in Caramoan, a tourism site known for its beautiful beaches, limestone formations, and karst caves. 3 important caves were chosen based on type of formation, accessibility, and access by humans. The first cave site is Kulapnit Cave in Barangay Tagbon, which is nearest to the town center and about 500 meters from the main road. It is also frequented by tourists who come to visit the Caramoan beaches. When we took our samples from the caves in March of this year, this was the first time that people had entered the cave in 2 years since the pandemic lockdown. Here is the team sampling from cave formations. And here are examples of the speleothems. The picture in the far right is the cave entrance. Another cave site was the cavernous Luyang Cave. This cave was the most inaccessible among the caves sampled and required a 4-kilometer hike up the mountainside from the nearest barangay. Even the locals rarely visit the cave because of its inaccessibility. Here is the team taking samples from the cave. Here are further examples of the speleothem. At far right is the cave entrance. The Patagbelen Cave in Barangay Patagbelen was the last cave to be sampled. Like Luyang Cave, this cave is also rarely visited by the locals and tourists. Again, here is the team taking samples from Patagbelen Cave. And here are further examples of the cave formations in Patag Belen. A total of 24 samples of rock scrapings and surface swabs from stalactites, stalagmites, columns, walls, as well as moon milk and drip water were obtained. Back in the lab, these samples were plated onto B4 medium that qualitatively screened for urolithic microorganisms. 60 isolates were identified to be urolithic. These isolates were then quantitatively screened for vital components of MICP as shown in the figure on the right. The urease activity test, the calcium carbonate precipitation test, and the Congo red binding assay for determination of extracellular polymeric substance or EPS were conducted. EPS is also important in establishing biofilms as measured using the biofilm assay. Urease activity is measured through conductivity since the breakdown of urea creates carbonates and ammonia in iron form. Hence, the more ions in solution, the higher the electrical conductivity of the solution. Luyang Cave isolate Ly6Eb showed the highest urease activity among the isolates tested. In the calcium carbonate precipitation test, cultures are grown overnight in urea. The buildup of carbonates and the high pH in the culture immediately causes precipitation of calcium carbonate upon addition of calcium chloride. In this test, Ly6Eb and Ly2Na, two isolates from Luyang Cave, performed the best. The isolates producing the most exopolysaccharides as determined by the Congo red binding assay are Ly6Eb, Ly2N1, and Pg5PF. The formation of biofilm is advantageous for bacterial cells to stick together and adhere to surfaces such as aggregates during production of bacterial bricks. The best performing isolates in the biofilm assay were also the three best isolates which produced the most EPS. From a total of 60 isolates that were initially identified to be urolytic, 4 isolates were identified to be the best performing ones based on 4 screening methods. These isolates were retested for selection of the best bacterium, and from all isolates tested, Ly6Eb was chosen as the best isolate to proceed to mutagenesis. Mutagenesis was conducted to further enhance the capability of the promising isolate for MICP. Plates of different dilutions of the bacterium were exposed to UV light for up to 120 seconds at a distance of 50 centimeters from the UV light source. The plates were covered to prevent photoreactivation and incubated in the dark for 24 hours. This figure shows the urease activity and specific urease activity of the mutants compared to the wild type shown on the farthest left. All mutants except Ly6EBM1 shown in the farthest right showed significantly higher urease activity and specific urease activity compared to the wild type. This figure compares the urease activity of the cave isolates to our laboratory isolates SS4 and UA4 and to the Ly6EB mutants. Mutant Ly6EB-MFD, which was grown under optimized conditions, had a 60% increase in urease activity compared to the wild-type strain. Take note also of the urease activity of the mutant strain over SS4 isolate, which is 200 times more. This will be further explained in the succeeding section. I will now discuss the actual process of bacterial brick production. Experiments on current isolates obtained from Karamoan, especially the mutant isolate, are still ongoing and have not yet reached brick production stage. However, the lab has been able to create bacterial bricks from previous isolates. There are four major components for the production of bacterial bricks through MICP. The bacterial medium containing a 24 to 48-hour culture of the bacterial cells, nutrient broth, and urea. The cementation medium containing the nutrient broth with urea and calcium chloride, sterile water, and the mold with packed sand. Bacterial and cementation solutions are introduced alternately in the column in succession 3 times. On the third treatment, the effluent line is blocked to retain the solutions in the column. The columns are incubated for 12 hours at 25 degrees centigrade to allow the crystallization to occur before draining the solution and adding another set of treatment. The columns are treated twice per day with a 12-hour interval for 4 days. After treatment, the columns are washed with sterile saline solution and then allowed to cure for 2 weeks before removing the cemented columns from the mold. The first strategy was adapted from the methods of Dami et al. And Omoreggi et al. The molds used were 50 ml plastic syringes with the needles and the rubber stoppers removed. The plastic tip of the plunger was cut off to produce a flat surface for compacting the sand column, and holes were drilled on the flat surface to allow the entry of influent solutions. The mold setups were sterilized with sodium hypochlorite. The sand was sieved, sterilized 3 times at 1-day intervals before packing into the syringe column. Following many trials and revisions, and adjustment of calcium chloride and urea concentrations, the first samples of bioconsolidated sand were successfully produced. Portions of the cemented plugs were removed and observed under a light microscope. Calcite deposited by bacteria were seen between sand particles. To test the compressive strength of the bacterial bricks, sand blocks were prepared using the specified molds. Thin plywood was used for the mold and was cut according to these specifications. The plywood was coated and put together using packaging tape. 4 holes were drilled on the bottom of the mold. Sterile air-dried sand was packed into the mold and covered with Wattmann filter paper and a plastic screen to prevent disturbance of the top layer during the addition of solutions. The upper filter and plastic screen were removed after 1 set of treatment. Bacterial bricks were prepared using cultures of bacteria isolated from the UP Diliman campus grounds. Co-cultures were formulated by mixing 50% of each strain. All molds were treated with 3 regimes of MICP cycle. The bricks were allowed to cure inside the fume hood for 1 week at room temperature and were transferred outside for another week. The produced bricks were tested for compressive strength. The bacterial bricks were tested alongside 3 controls of mortar cubes with 5%, 15%, and 25% cement by volume. The bricks produced using co-cultures were weaker than the other samples produced using one type of bacterial isolate. Bacterial brick produced using UA4 was comparable to concrete block with 5% cement, while the brick produced using SS4 was comparable to concrete sample with 15% cement. With these initial results using strains that were isolated from around the campus, there is certainty that the mutant strain from Luyang Cave will be able to outdo the initial results for brick production, especially since the Luyang Cave mutant had a urease activity 200 times more than the isolate SS4 in this preliminary study. Other methods of brick production will be tried for the mutant strains, such as the surface percolation technique and the immersion method, which are the newer methods for MICP production. Other applications of MICP are in crack repair, recycling and improvement of concrete, production of self-healing concrete, dust suppression, restoration of limestone structures, and bioremediation of heavy metals. This is indeed an exciting and emerging field. The authors would like to acknowledge Dr. Mike Flores of Partido State University in Caramoan for facilitating the sampling, and the Department of Science and Technology, Philippine Council for Industry, Energy, and Emerging Technology Research and Development, or DOSTP, shared for funding this project. Good day, everyone.
Okay, thank you very much, Dr. Jessica Simbahan. So we will continue to our next presenter. Now we have a presentation from Professor Dr. Benjamin Charslip from Prince of Songkla University, Faculty of Agroindustry, Center of Excellence in Innovative Biotechnology for Sustainable Utilization of Bioresource from Thailand. And the title of the topic today is Biovalorization of Agroindustrial Waste as Low-Cost Nutrient Source for Organic Acid Production. So we will hear the presentation now.
Good afternoon, everyone. My name is Benjamin Cheng Sin. Today, I would like to present the research of my master's student, Ms. Asama Bilate. The topic is biovalorization of agroindustrial waste as low-cost nutrient source for organic acid production. The demand for lactic acid is expected to increase due to new processing technology and further field of application of biodegradable polymers, especially co-crystallization occur when high optical purity of poly-L-lactic acid is mixed with the poly-D-lactic acid, resulting in a new stereo complex with higher thermal stability. Lactic acid production can be achieved either by chemical synthesis or fermentative production. By chemical synthesis, a less mixture of the L-lactic acid is usually produced. On the other hand, fermentative production offer advantage of utilization of cheap renewable substrate, low production temperature, low energy consumption, and mostly production of optical pure D or L lactic acid when the appropriate microorganism is selected at the lactic acid producer. On the other hand, most lactic acid species require complex nutrients, including amino acid, peptide, nucleotide, and vitamin for their growth because they lack many biosynthetic capabilities, which hamper the recovery of the lactic acid and increase the production cost. Thus, the use of low-cost nutrient source has been extensively investigated. In this study, we are interested in one of the most interesting agricultural materials in Thailand is oil palm trunk. As a sap form, oil palm trunk contains large quantity of high sugar and also nitrogen and vitamins. It was considered as a low-cost nutrient source for microorganisms. In addition, oil palm trunk after squishing contains a large amount of cellulose and hemicellulose, so it can be divided to vascular bundle or VB and parenchyma or PA is have different compositions, and both of them can be converted into fermentable sugar and also lactic acid. So they could then be promising low-cost nutrient source. So this study we designed and explained from the upstream to fermentation and downstream. Let me explain for the upstream. We isolate and screen the D-lactic acid and L-lactic acid producer. In this study, we isolate lactic acid from oil palm sap itself and also from the kefir grains. So that has been reported that they contain the D-lactic acid producer. We spread it on the MIS agar, and we select the colony that exhibit the yellow colony and restrict to purify, and then it will test in catalyst and gam staining. So the strain that show catalyst negative and gam positive will be selected. In the primary screening of high lactic acid producing stain and detection of the isomeric forms, we found 7 isolates from the SAP and 27 isolated from kefir grains. We also compare with the reference stains. The result of a primary screening show that only 5 stains can produce the total acid yield higher than 0.8 gram per gram sugar, and only 2 stains are able to produce D-lactic acid as a major isomer. The newly isolated strains were then identified. 16 sRDNA fragment of the isolate strain were amplified using the universal primer, and the sequence was brushed, and with the 100% similarity, we can identify the newly isolated one to be Lactobacillus paracasei and Lactobacillus kefiri. And in summary, the design stains that obtained from part 1, so show in this slide. So we have 5 stains, and then we use this one for cultivation in the oil palm sap. So this is the second part. We start with the screening of the lactic acid bacteria using the oil palm saps under pH control and un-pH control. In this part, so 5 stains were cultivated in the oil palm sap, and then we compare the lactic acid production. So this slide shows the time course of the disulfide and lactic acid production by the lactic acid producing bacteria. As you can see, the strain that we newly isolated one is produce high biomass and also the high lactic acid compared to the GCM 6985, and it's also consumed the total sugar faster than the reference strain. And when we cultivate under the pH control, so the cell growth was much improved, and also the kefiri can produce higher lactic acid, and the total sugar was consumed rapidly. And this is the skinning of the L-lactic acid producing bacteria. So all stains grew similarly, but only the Lactobacillus acidophilus STR1338 can produce a much higher lactic acid. And under the pH controls, the lactic acid was more improved because we can reduce the acidic pH inhibition. And this part, we try to increase the carbon salts for the lactic acid bacteria. So we also add the oil palm trunk fiber after squishing the sap already. So we use the Vesela bundle. So we use the OPT-VB. So it's more fiber, it contains more cellulose and hemicellulose compared to the parenchyma. And so we use the simultaneous certification and fermentation of SSF process, because this one can reduce the reactor volume, and it can reduce the processing times, and also reduce the feedback inhibition and increase the productivity. It's because we can do the saccharification and fermentation in one reactor. However, in many cases, for the SSF, the p hydrolysis step is required in order to achieve considerable amount of sugar for initial growth of the microorganisms. But in this study, as we use the OPA, so it already contains certain amount of sugar and nutrients, so it can be used directly as the initial nutrient salt for the lactic acid bacteria. We don't have to do the p-hydrolysis. And in this part, we use only 2% of the representative for the D lactic acid producer and L-lactic producer to compare the lactic acid production. And in this section, so we vary the amount of the OPT that we add in. So from 5%, 10%, and 15%. And you can see that 15% is the maximum that we can add it. And then we do the cultivation, and then we compare the lactic acid production. So this is the time course of the total sugar concentration and total acid. So we can see that the sugar concentration increased because the rate of the saccharification is faster than the rate of the fermentation during the initial 24 hours. But after that, the cell grew rapidly, and then they consume sugar to produce the total acid or lactic acid. So when we increase the OPT, so we can get more lactic acid, but actually when we compare the result between 10% and 15%, so the lactic acid use is not much improved. This might be for the mass transfer limitations, and it should be noted that this newly isolated Kefiri strain also produces acidic and ethanol at very high concentration, so it's a heterofermentative. And this is the result for the L-lactic acid production. So we have this similar trend of the total sugar concentration and total acid. And the use of the OPT at 10% and 15% did not give much different lactic acid production. So we choose the 10% OPT to be added in the system. And this one is the study of the effect of the enzyme loading. So we vary the enzyme for the cellulase at 5, 10, and 15 and 20 filter paper unit gram per gram OPT. This is the time course. So the figures show that higher enzyme loading led to higher sugar release, and is indicate the rapid enzymatic hydrolysis in the initial period, and sugar releasing was faster than the sugar consumption. Obviously, a higher enzyme loading can promote more lactic acid due to the faster sugar release, but the excess enzyme loading did not improve more sugar release and lactic acid production. So we choose the minimum one at 10 filter per unit per gram oil palm trunk. And this is for the L-lactic producer. So we also got similar result, but for the L-lactic, so there is some difference between the 10 filter per unit and 15 filter. So we choose the 15 filter per unit for this strain. And when we compare the lactic acid production by 2 strain, we found that the lactic acid acidophilus, lactobacillus acidophilus TISTR123 have much higher lactic acid production, so it was choosing for further study. And in this study, we proposed for the repeated SSF as the lactic acid was accumulated in the culture box, and the pH the medium was dropped, so resulting in the end product inhibition and also acidic inhibition. So in this study, we do the repeat simultaneous saccharification and fermentation. So after finish the first batch, so we replace the fermented batch with the new OPS and also the enzyme, and then but we still use the remaining of the OPT. So with this strategy, we can effectively use the OPT for 5 cycles. And then when we compare the solid residue, so you can see that the solid residue decrease until the batch number 4, and then the total sugar also during the fermentation, so the total sugar what released by the enzyme hydrolysis, and then is what consumed by the lactic acid bacteria. And then you can see the total acid for each cycle. The maximum one we got at the batch 2, so we can repeat for 2 or 3 batch. And after that, because the cell is already inactive, so we got lower total acid production, and also because the OPT is already mostly already hydrolyzed, so we got less carbon salt. So I think the 3 cycle is most suitable for the use of the OPT with the OPH in the repeated simultaneously saccharification and fermentation. Okay, you can see this one. Okay. And this is the comparison between each batch. So even though the lactic acid production reduced, but in the 5th cycle is still high. But the highest one is in the 2nd batch. And after that, we scale up the process from the serum bottle to be 1 liter. And when we also repeat the experiment, and we found that we can produce comparatively high lactic acid production, even though in the 1 liter reactor. And this part is the use of the parenchyma. Is the different from the Vasella bundle because it contains a lot of starch. So in this study, we also add the amylase, and we found that amylase also help to increase the total sugar and also the total acid. So it's also possible to use both part of the OPT as the carbon salt for the lactic acid production. And the last part is the lactic acid recovery. So in this part, we study for the resin loading, the absorption isotherm, and we also do the desorption of lactic acid. And actually, we study with the pure lactic acid first, and after that, we use the fermented batch from the 1 liter of the experiment. And before do that, we have to decolor the color of the culture board. So we use the activated carbon first, and after that we can get the clear spermatan, and then use that one to recover the lactic acid in the column. This is the result. We found that when we increase the resin loading up to 20%, so we can get the maximum absorption, but we cannot add more resin because it's already absorbed the liquid. And for the isotherm study, we compare between the Langmuir and Friedrich, and we found that Friedrich is more suitable for the absorption of lactic acid on the resin in this study. And after we get the saturated resin, we also do the desorption by the ethanol, and then we can recover. Actually, so we can recover the resin and also separate the lactic from the resin, and the resin can be reused. And when we do the column, so we can absorb until it's saturated at fraction more than 20. And so the absorption percent is only a 40%. And so after we do the decolorization, we also use the fermentation board to recover the lactic acid, and we found that the absorption is slightly decreased. It may be because of other compounds in the fermentation board. So we have slightly decrease of the absorption. And when we compare the lactic acid absorption by the activated salad, we found that only small amount of lactic acid was absorbed in the activated carbon. So it's possible to use the activated carbon for the decolorization. And so in this study, I would like to conclude that by overalization of OPS and OPT at co-substrate for lactic acid production through simultaneous saccharification and fermentation was successfully performed. The repeated SSF and feedback SSF was effective for efficient utilization of the OPT. The recovery of the lactic acid using strong base anion resin show absorption isotherm closely predicted by the finish. And the lactic acid can be recovered and resin can be used. This study has provided a promising strategy for efficient use of the cellulosic agricultural waste for production of organic acid. And that's all for my presentation. Thank you for your attention.
Thank you for presentation. So from Professor Dr. Benyamas, we will continue to our next speaker, Dr. Paul John L. Geraldino. Dr. Paul is from Department of Biology, University of San Carlos, Philippines. And the title for today's presentation is Fermenting Seaweeds to Improve Poultry Health. So without any further ado, we will hear the presentation now, please.
Hi, I am Paul Geraldino from the Department of Biology, University of San Carlos, Cebu City, Philippines. I will share to you our research work on fermentation of seaweeds to help improve the health of chicken. This work is an ongoing work and part of a collaborative research with a Taiwanese group in Taipei. Seaweeds have been used for centuries as feed supplements in livestock diet. For example, ancient Romans fed horses with seaweeds. Also in Iceland, Norway, France, domesticated animals were fed with macroalgae. This is because seaweeds possess high-level minerals, vitamins, non-digestible polysaccharides, dietary fibers, high-level proteins, low level of lipids as compared with terrestrial plants. The benefits to livestock fed with seaweeds showed improved growth performance, quality of carcass, resistance to disease, and add to nutritive property of the meat. We are actually working closely together with academician Dr. Coke Montaño, guiding us on what seaweeds to study. You see, the diversity of seaweeds in our country is actually high. There are about 893 identified seaweeds according to Dr. Put Ang in 2013, and I bet there are still a lot of species to be discovered in our waters, including their potential uses and application. That's why Dr. Koch is always pushing us to continue working on seaweed research and innovation. At the University of San Carlos, we have a marine research station in Mactan Island, wherein Dr. Alvin Monotilla, one of our younger colleagues, is rearing and maintaining seed stock culture of the green seaweed ulva. These species of seaweeds are abundant, especially during summer months, and even causes green tides in most part of our intertidal seas. But remember, seaweeds in general should be mixed with animal feed because when consumed alone can cause negative impact on animals. So our team decided to work on fermentation or bioconversion technology and work our way into developing animal feeds using seaweeds. As background, fermented feeds refers to the degradation of macromolecular substances in feed into small molecules through microbial metabolism, perhaps to improve the digestibility of macro and micronutrients. And more recently, studies have shown that fermentation have been associated with methods that improve the nutritional value of unconventional feed ingredients for broiler chicken. And in order to further improve the fermentation process, we encourage to follow the strategy mentioned in reports, which shows that polysaccharides from seaweeds that are unsuitable for fermentation can be overcome by degrading first the cellulose of seaweeds before proceeding to fermentation process by simply adding bacteria or yeast to enhance the fermentation process. Our team in the department has been maintaining culture strain which are economically important and have biomedical and agricultural applications. Dr. Juni Yi, my colleague in the department, is our point microbiologist of our group. We have been isolating microbes from inhospitable areas to areas within our campus. Most of our strains we characterize chemically and physically, including molecular verified identification. And so we have access to several microbes that can best ferment our seaweeds of choice for our current study. So one of the objectives of our current research is to develop a feed additive formulation to supplement the growth and immunity of poultry chickens. We continually search for more microbes from the field and regularly check to see if they can efficiently ferment specific seaweeds. And there are two fermentation methods that we use in our laboratory. First, solid state fermentation, and second is submerged state fermentation. We optimize the fermentation conditions, not just one condition, for each of the seaweeds that we tested. So for this talk, I will share to you our research result on these three seaweeds. We work first brown algae padina, one of the few calcified brown algae, sister to sargassum, green algae ulva, the one that we cultivate in our marine research station used as condiment in Japan, and red algae halimenia, which is actually abundant in the Visayas region and are consumed as delicacies in some coastal communities. It is important to consider that when including seaweeds in animal feed, their composition is always variable, which depends on the species, habitat location, harvest time, growing conditions such as nutrient concentration in water, light intensity, temperature, etc. So therefore, during the drying step, we check the proximate and mineral composition regularly. We usually compare our test results with outsourced service facility also, and oftentimes the value vary, but not really that drastic. Values are not that far, but we monitor closely the moisture content of our product. We are currently doing laboratory scale fermentation, optimizing every formulation that we develop before recommending to industrial scale level production. We have been screening few species of lactobacillus from our collection. Here you can see a very simplified protocol for fermenting seaweeds. And if you noticed, we actually made use of crude cellulose enzyme that we also developed in our laboratory as an important step in fermentation process. The crude cellulose enzyme in our laboratory was an extract from a couple of bacillus species that we isolated from a specific species of termites. Actually, we isolated several species of microbes from the gut of the termite, but these two are the ones exhibited the highest cellulolytic ability. Also, we found that in most cases, submerged state fermentation is easier to maintain and monitor compared to solid state fermentation. Most of the seaweed microbe combination we tested performed better in submerged state fermentation. And among the three species of seaweeds, we tested the brown seaweed padina first. Despite the test, chickens eating all different supplements quite happily. So as you can see, compared to the control, the highest mean body weight is when using 2% seaweed feed additive. We compared also the fermented seaweeds with the unfermented dried seaweeds because again, these are already usable for any animal feed inclusion. Here, the brown seaweed padina showed relatively high mean total weight response from our tested chickens. Still up to now, me and our team is continuing the optimization of the fermentation step to make sure we find the perfect balance. And we realize that the drying step is quite an essential step in the preparation for fermentation. We also compared the mineral composition of the formulated feed supplement in every batch. Here you can see the variability of the seaweed products. As I mentioned previously, composition is not the same every batch. So again, the biomass and the collection source plays a crucial role in optimizing the product development process. Therefore, what we did, we established several coastal community partners where we usually get our biomass just to be consistent as much as possible. The feed conversion ratio among different diet treatments in this set using fermented padina are again comparable to the control. I believe in most of our trials we get very minimal fatality, like 1 or 2 per batch. All the chickens are eating and growing very well within our facility. So I think we have a good set of veterinary technicians who closely monitor our baby chickens. Feeding trial is the part where we had the most difficult time because we are not an agricultural school. Although we have in-house veterinary medical doctors, but we still needed help from the expert in the Department of Agriculture Region 7. And also we have partners with few local poultry growers and gather expert opinions in order to guide us in this testing step. We also examined the carcass and tried to see some differences in the meat quality. There were no clear winner, but generally the quality of meat is very good and no particular diseases were detected. We are doing a few more physiological and histological analysis in the laboratory now to further test the health benefits of the fermented seaweed feed additives or supplements to the chicken. One noticeable immunological response of our chicken during the feeding trials is the elevation of the immunoglobulin Y blood serum. We do this every test batch and few other blood chemistry screening to check the health immunity of our chicken. According to studies, the total serum IgY concentration could also be used as an index for offspring fitness because of the positive correlation between total IgY concentration and overall chicken health. So this is my last slide, and again, there are beneficial applications of seaweeds, including their extracted compounds, which have excellent health improving properties that can be utilized as feed additives, with which can impact animal health and production. So yes, again, our work is still ongoing and there are still much to do. This is why I would like to thank the DUSTP card for the continued support and for the grant given to us through Mekoteco program, and to our Taiwanese collaborators from Furen Catholic University in Taipei. Thanks also to my research team, to Dr. Juni Yi, Jem Rania, Herman Tan, our colleague in the department. Thank you for listening.
Okay, thank you very much. So we will come now to a discussion or question and answer session. For the audience who want to have a question, you can write your question in a chat box. So meanwhile, we waiting a question from the question. So I would like to have a question for our first speaker, Dr. Jessica. So in your presentation, there is the bioconcrete. Yeah, it very interesting. We make it from the help with the microbe, but do you think is there any concern regarding maybe volatile agent that produced by the microbe that probably have a toxic effect when meanwhile they make the bioconcrete? Is it necessary to assess that? Sorry, maybe you can unmute your mic first.
Oh, I'm sorry. I'm sorry.
Yeah.
That's a very good question, because one of the what's this, byproducts, as you can see from the process that I showed you, is ammonia. So there is a need to find a way to recycle the ammonia, and that is being looked into, as well as, yeah, I think ammonia is a major uh, uh, cause for, not a cause for concern, but it has to be addressed. The Because there is ammonia that is released from the reaction. Yes.
Okay. Yeah, yeah.
And
And another thing, yeah.
recycling
Yeah.
is also one of the areas for research.
Yeah. It's very interesting. And another application that also you show in your slide, it like can be for sealing, yeah, sealing maybe the broken concrete, yeah. So,
Yeah.
in your opinion, if it will be applied for that application, yeah. So,
Yeah.
because you said that it needs several time or days for the concrete to be made by the microbe. So, because the concrete will be in outside, yeah, in the environment. So how could we maintain the condition so the microbe later on can seal the concrete?
Yeah.
I hope you can understand my question. Yeah.
Yeah. I think you mean crack healing. For
Yeah.
healing of cracks. Yes. There are different strategies for that. And one strategy is to incorporate the microorganism into the cement before you even put up the structure. So that when the crack appears, then the microorganism, once it's exposed to the crack, you will have water, carbon dioxide, and all that. Then the microorganism can already do its job. Yeah. So there are also strategies to pelletize the microorganism to give it already the nutrients it needs to grow once the crack is made. And then the microorganisms they can be placed when the crack is formed, or they can be incorporated already when you make the structure. Did I answer your question?
Yeah. Actually, it's
Yeah.
very interesting because in nature, actually the microbe also how to say, can be live without our help. Yeah. So because the nutrition is already provided by the nature. Yeah.
Yeah.
So it's very interesting actually, Dr. Jessica.
Thank you.
Okay, thank you. So maybe I would like to address my question for Professor Benjamas. So for your presentation, the bacteria that you able to isolate, is it lactobacillus, or is it another strain of the lactobacillus?
Actually, it's the lactobacillus. Yeah.
So for this because you said that we can how to say utilize the waste from the industry, agro industry. Actually, is it the same like that my question that I answered for Dr. Jessica? Is there any concern for the safety if we use this the organic acid? I don't know whether it can be consumed directly or it should be utilized for other application.
Actually, our aim is to produce at the feedstock for the biopolymer. Is the polylactic acid. It's a biodegradable polymer feedstock.
So by using this bacteria, so we hope that we can produce the biodegradable polymer. Yeah. Is
Yeah,
it correct?
so we will produce the lactic acid first, and then the chemistry have to use that lactic acid as the raw material to produce biodegradable polymer later.
Okay. So
Yes.
we will not use directly the organic product. Yeah.
Yeah, we cannot
Yeah.
use it. We have to convert it by the chemical method again to the biopolymer.
Okay. And
Yes.
in your study, is it the bacteria that you found, the strain, is it how to say, it's more powerful rather compared to other isolate?
Actually, they can produce the D-lactic acid bacteria, and we also got the one that can produce lactic acid bacteria at the comparable high yield compared to the reference strain. And especially for the D-lactic acid producer, is grow better than the reference strain.
Yeah, so it's very economic. The economic value is very high, I think. Yeah.
Yeah, we hope so because we use the same.
Is there any industry that already interesting with it?
Yeah, actually, we start from the company that who want to make use of the oil palm trunk. Oil palm trunk, they just ask us that how can we convert this trunk to be the valuable products? And as we are biotechnologists, so we just try to use the microorganism to convert it into the lactic acid. That one is the feedstock for the polymer because we think that the biodegradable polymer is very high demand in this day.
So in Thailand, is it already common to use such a technology in the for the waste solution?
Yeah, we have a lot of proposed to use the agricultural waste for the microorganism to produce the bioproduct. But for the oil palm sack, is not yet developed. So I hope that this one would give the roadmap for the company to use the waste for the lactic acid production in the future.
Yeah, because it's very interesting. I think Thailand, Malaysia, and also Indonesia is very high in producing oil palm. Yeah.
Yeah.
So it's very, very interesting presentation, actually, the Professor Benjamas. I'm
So
really
much.
appreciate with this. Yeah.
Okay.
Okay. I think we have no Dr. Geraldino with us here. Yeah. So I think maybe because there is some matter, so he cannot join with us. But before you all how to say finishing the meeting, so actually committee will ask all of audience to back to the main room, so we will have the closing meeting in a main room. So I think because our session will be end now, so we will have the awarding certificate for our presenter, and after that we will ask you to make a photo session together, and then after that we back to the main room. So before that, I will ask a committee to show or display the A certificate for our presenter. So first, the first one, we will address for our first speaker, Dr. Jessica Simbahan. Okay, thank you very much for your inspiring sharing.
Thank you, thank you also.
Yeah. Okay, and the next one will be addressed for Professor Dr. Benjamas. Okay, thank you very much also for your, I think it's very, very a big impact letter if we can apply this, yeah? Thank you very much.
Yes.
Yeah, the last one, we will award this for Dr. Paul John Geraldino. Even though he not come with us now, but I think the presentation also inspiring for us. So thank you very much for everybody. I hope we can open up the camera so we will have a photo session together in this meeting room. Can you open up your camera, please? So the committee will make a photo session for us. Maybe committee can. Okay. So everybody be prepared and give your very, very good smile. Okay. So, okay, we have a photo. So the committee, is it okay? Okay. We have 2 slides now. Yeah. Yeah. Okay. Okay. Do we have all the picture? Okay. Thank you very much for everybody. Now we can join to the main room because we will join the closing ceremony in a main hall. And in a main hall also, we will have some questionnaire to for the committee to have your opinion regarding this event. Yeah. So we will back to the main room. So please back again to the break for the main room. Yeah, we see you all in main room. Bye bye.
Recording stopped. Recording in progress.
So thank you very much for the distinguished guests and the honorable speakers.